I suppose we're still in January of 2023, so it's okay to say Happy New Year. Welcome to everyone, and welcome to, I will say, season 2 of Tuesday Talks. We launched Tuesday Talks last year, and it's a way for us to build community here at Cure. And it's a pleasure to actually kick off 2023's first Tuesday Talk, and we have a really terrific topic. I don't know how many of you actually know about Cure and or are residents here, but just a few words. First of all, Cure is an affiliate company of Deerfield Management, and our speaker today is, of course, from Deerfield, and Cure is an innovation campus. We call ourselves an innovation campus. We have world-class laboratory facilities, medical device incubators, and incubation collaboration residency, but also an education center and space like this, because really our goal is to transform healthcare and bring forward cures, cure period, as you can see from our logo, and that is by, Cure can't do it alone. It takes more than a village, it takes more than a community, it takes an entire ecosystem in New York and beyond to actually transform healthcare and find new cures. So that's our goal. And today's topic actually is terrific, and it comes from the parent company Deerfield and someone from Deerfield who's been a thought leader in this arena, and that is the reproducibility problem. And we're going to hear a lot more about it and how it impacts drug discovery. And to actually take us through this is Christine Brideau. She is actually the vice president for preclinical pharmacology at Deerfield Discovery and Development, that's 3DC. She joined the firm in 2019, and before that, she was vice president of in vitro pharmacology at WuXi AppTec, and she led the development of WuXi's US-based in vitro biology unit in New Jersey. And before that, Christine spent 24 years at Merck while I was at J&J, you were at Merck, right? And there, again, most recently served as executive director of the in vitro pharmacology and supported multiple drug discovery projects from hit to lead to, clinical stage, right? And that must have been just an extraordinary experience. And you've been the co-author of more than 40 peer-reviewed publications in the area of pharmacology, assay development, screening, day analysis, automation, compound management, and more. I mean, drug discovery is a really complicated process. A lot of people don't know, how iterative it is and how many stages you have to go through before you actually have a hit and a lead, right? And you're currently the editorial board member of SLAS Discovery and served as a reviewer for the NIH Molecular Libraries Program Study section. So, Christine, you have so much to talk about today, and the format that we're going to follow is just to set the context. Christine's going to walk through a few slides and talk us through just this process of drug discovery and assay development, and then what is this problem of reproducibility, and then after a few slides, then I'm going to do a moderated conversation, and then we'll open it up to question, because this is a really important topic. Not really, something that people speak a lot about, but it's an important thing for us to find solutions. And the good news is that a lot of really, great minds are thinking about how do we solve this. So with that, Christine, welcome. Thank you.
Great. Thank you, Seema. Thanks for inviting me on and to talk about this. It is dear to my heart because it's something that I have been living with for many years, and I wanted to share from the perspective of the industry. I worked at Merck, as you heard, and now that we're working here at Deerfield, we're facing the similar issues, and it's something that we're constantly struggling with. And I think we need to discuss and open up about the reproducibility problem. This is something that has been surfaced about 10, 12 years ago, and so I just wanted to put it maybe in context so that we can kind of focus the discussion on something that I can talk about. I'm a pharmacologist by training, and I'm working more in the preclinical discovery side. So this is an area that I'm more comfortable and I can answer questions on. So what is the reproducibility problem and its impact on drug discovery? And we go through the disclaimers. But just to recap, in my world of drug discovery one is understanding the target and the disease indication and how we can develop a therapeutic for that target and disease. And it often starts with target validation. We start with experimental models, genetics, or in cellular models. And then once we have an idea of the target and how we want to develop a drug, we then have to do compound screening. And I have more experience in small molecules, but we also have biologics, and it's very similar. We have to develop assays that we can screen through all these compounds or biologics. And once we've identified some prototypical lead compounds, we then have to understand the mechanisms. We have secondary assays, eventually test in an in vivo model, and then go to the clinic. There are a lot of steps. It takes a lot of effort to do this kind of work. And in our minds, right at the beginning, we understand that the goal is to go to clinical trials and understand that you're going to give this drug, potential new drug, to a patient. This patient could be your family member. I have had the privilege of having my father taking a drug that I developed from Merck. So keep that in mind. Then, in terms of the drug discovery, and it is an iterative cycle, and decisions need to be made at every point of that circle. You need to understand the data, analyze the data, design the molecules, make the molecules, test them, and a huge team of scientists and experts are involved in making these decisions. And it requires robust data to make those decisions. And what we have been struggling with is, at the beginning especially in industry, we're looking for new targets, novel therapeutics. And we read the literature. A lot of our ideas come from the literature. And for some of us, I think many of us have had the experience of not being able to reproduce the data. And then you tend to put it on yourself. You say, I didn't do it quite well. And then, lo and behold, about 10 years ago, more publications came out from Bayer, then Amgen. They were the first ones to really publish and recognize that this is a problem that's pervasive. And then Nature just came out with a survey. They surveyed 1,000 scientists, and even the scientists, about 60–70% of the scientists said, yes, I've had that problem where I couldn't reproduce the data. So it's quite pervasive, and I think it's time. Well, a lot of discussions have been going on because of this, but I think something we should continually discuss and try to improve and find solutions or suggestions on how to address this problem. So that's to set up the context.
I think that's a great setup, Christine. I mean, any one of us who spent even a little bit of time in science, right? We all know that one of the foundational pieces, the bedrock of scientific research, is reproducibility. So that's what gives it credibility, the fact that you can reproduce it. And yet here we are facing this problem. But talk a little bit about reproducibility and the importance and why that matters. Why is that so important?
And so what we have found is there's a lot of wasted time and effort on working on something that perhaps wasn't strong enough hypothesis, and the results really are not strong enough to support it. And what the frustration is that in industry, we spend a lot of time initiating projects and spending time and money to then realize there may be not a path forward. Sometimes there is. Sometimes we can find alternative methods, but at the end of the day, it ends up wasting time. In fact, there was a study about economics of assay reproducibility, and in the US alone, in terms of the life sciences and drug discovery in general, which includes governments, industry, they're saying about $56 billion is spent on this effort. And based on these publications, about 50% is not reproducible. So they're claiming that $28 billion is potentially lost.
Wow.
It sounds incredibly high, but it just gives you some context on how important it is.
Yeah, but it's also taking a step back, right? The ability to reproduce is the way you create, credibility that this thing is going to work, ultimately, correct? So what's the solution? I mean, how do you kind of do that? Are there steps that you can take, things that you can do to make sure that you're not actually losing that much time and wasting that much effort? How do you do that?
So one thing that we do in industry is try to get as much information and detail as possible. But we all end up, and I think now, and I was just talking to some colleagues earlier, where I'm from Merck, but others are from Lilly and BMS. We've all started, whenever we start something new, we try to replicate what's been done from the literature. But I think and that's what we typically try to do, is really try to understand where are the weaknesses, where are the strengths. But ideally, we shouldn't have to do so much effort.
Yeah.
That would be ideal, not to have to do that. And the lack of protocols, detailed protocols, lack of good reagents. The reagents are a big issue.
Yeah. So who does it impact this? I mean, is it really something that sort of stays in this preclinical environment? The drug discovery scientists who are, spending so much time and effort trying to find that, that hit, maybe the big, gold mine, or does it really impact things down the value chain?
I do think that it does impact not only the upfront, but down the chain, and it actually impacts the patients.
Yeah.
If you're not able to, the industry is fast moving and don't always have the time to spend on getting something into a pipeline, and things may stall and decisions may be made not to pursue. And that's a lost opportunity. Maybe we could have developed something for the patient. So there's the patient, there's also the cost of the drug development, because if you add up all of the time and effort, it adds to the cost of drug discovery and development.
So that cost, of course, gets, passed along ultimately in the cost of the drug and therefore the patient.
Oh, well,
right?
We know that drug discovery is really hard and most things end up in failure. So if you can start off with a high probability of success, I think that does result in
some
cost savings.
So, it just occurs to me, I mean, high throughput screening was all the hype, right? Because once the genome was sequenced, then we could just, quote unquote, do a lot of, screen a lot of compounds, right, in one fell swoop. Has that contributed to it? Was it different before, before all of the high throughput screening, where you were just looking at the gels and well, that was a long process.
I'm going to age myself. So I started in industry at 1990. And in the 90s, most of the internal research in pharma was done within pharma. Academia was basic. And so we did a lot of our own internal work, and the validation was different because we had a different goal. But with the advent of high throughput screening, I was part of the MLPCN effort, the NIH effort, where high throughput screening was opened up to academia, which was great. The idea was to have the opportunity to allow academia to have tool compounds, because that was always a problem.
Yeah.
Academia couldn't access pharma's libraries for finding hit compounds, so it's like, well, let's open it up. But I think that created also inadvertently some issues as well, because this is an area that if you need expertise, you need to understand how to discover small molecules, and that was the idea of the NIH with these probe endeavors, is really being careful about what you're using as your tool molecule for validating a target.
Yeah. And, you know, of course, the hit to lead ratio, you know, it became more and more and more and more important as time went on. Now, so in terms of reproducibility, are there any sort of rules or systems in place that, you know, tell you, well, it has to be reproducible before you can actually take it, you know, to the next step beyond discovery and into preclinical and then into real, you know, first in human?
Yeah, I think when it comes to the industry, we have to set in a lot of rigorous validation early on. So whatever is not meeting that criteria will be weeded out, and eventually, once it progresses into the later phases of drug discovery and development, it's a little bit less, I should say, the probability of successes are higher. But there are issues along the way anyways. I was talking to a colleague of mine who's in CMC, and she was saying, you know, even though we're in development, this is still an issue. So it's still an issue across every discipline. And again, it's about having that rigor and the discipline and being able to communicate the details of protocols.
But I'm thinking that this is even more of a pronounced problem in biology,
Yes.
rather than, let's say, in physics or chemistry, right?
Correct.
And why is that so?
In biology I think it's so complicated. The experiments that you set up in biology are very complex. You're having to create a lot of reagents. You're using a lot of technologies. It's getting more complex, the types of technologies that we're using. And in academia, what I've seen is you have these big labs, and we have postdocs who are working on projects, but the projects may last beyond the lifetime of one particular postdoc, and the information transfer and technology transfer from one student to the next is, you know, perhaps lacking, and it is complex. I mean, it is a problem, and it's hard. But I think the nature of the biology work lends itself that it is probable that something gets missed along the way.
Yes. And you know, and that's why the so-called digital, you know, industry has not yet been able to crack the code, because biology is complex, it's messy, you know, it's not like physics or chemistry, right? Can you give any examples of where this has actually sort of been shown as a problem that we as, you know, an industry need to like take a look at and solve?
Well, you know, speaking from my personal experience, it's really frustrating because we all want to succeed. The academic lab have some really interesting findings and want to progress it, and we're trying to use that learning and to progress it. And we all want a shared goal. We do want to discover a drug for the patient. But where it gets challenging is like little simple things. So I have one example when I was at Wuxi, actually, when I was working with an academic lab. And it was a very compelling target. It was, they had set up these cell lines that we could use to develop drugs for. And and we said ahead of time, can you make sure that your cell lines are not contaminated with mycoplasma, so that we don't want to receive dirty cell lines in our labs. We don't want to contaminate. So just make sure that your cell lines are clean before. Lo and behold, they were contaminated. So a postdoc spent 2 months cleaning up the cell line, and at the end of the day, could not reproduce their own data in the lab, and everything went dead.
Came to a grinding halt.
Grinding halt.
Yeah. And that's a lot of time and effort, and you never know. Maybe it was a good target, but because you couldn't reproduce it, then
Exactly.
Yeah. Are there examples of where something looks great and you keep going and then it fails in clinicals?
Well, there is a famous story, and I don't know if many of you remember this, when GSK spent $720 million to buy Sertris. This was based on red wine, resveratrol, and it was found that it would prevent aging. It basically recapitulated calorie restriction and anti-aging effects. So big news. And GSK bought this company based on this, and they weren't in clinic yet, but it did go to clinic. But what was interesting is that other companies like Pfizer and there's another one I can't remember right now, came out and published, and they said, the assay that you used is an artifact. Oh. Your compound does not bind to sirtuin-1 at all. It's just an artifact of the assay. It's actually binding the substrate and quenching the fluorophore. Wow. So this is an impact of one assay and a compound that really did not have any effect.
And in the meantime, we all bought cases and cases of red wine.
Red wine. I still drink my red wine.
Yes, I still drink mine, yes. Irreproducible or not, red wine is good for you, is what we believe. Yeah. So I'm sure that there are many people who are thinking about this problem and trying to come up with solutions, because, it is something that is not well known, not talked about, but, good minds, must be working on this.
So the NIH is on this.
Good.
This caught the attention as soon as all these papers were coming out in 2010, 2011. The NIH started looking into this, and now for most of you who are doing grant applications will know this, but they've instituted now a rigor and reproducibility policy, and there's more training. And I think a lot of the issue is that there's so much pressure on academia to publish, and it takes time to do things, to write things down, to write detailed protocols, to make sure the reagents are good. It just takes time. And I think what NIH is trying to train new scientists is just the discipline to really make sure that you have some rigor in what you're doing, and just making sure that you have validation along the way, and that you have right protocols.
And then what about in the future? Because I know we keep thinking about, are there ways that, technology can help to actually not just speed up the process, but to actually identify, maybe AI, identify some steps that, human error went wrong, and,
Well, the one problem I have with AI is that if it's using public data and it's literature data, you kind of have to wonder what's going to come out of it. I'm not sure how AI, maybe AI can help us identify holes in protocols. When we're reading a paper and we go to the lab, we're looking at the description of the protocol, and sometimes there's pieces missing. Can we go to the computer and tell us, give me a protocol to do X, Y, Z, and maybe AI could help with that? That's just thinking at the top of my head, but that might be useful, is that can you leverage AI for the different protocols that were done so that you can, even when you're cooking and you're doing at home and you're following a recipe, I don't know if some of you do this, but I look at multiple recipes just to see
and
mix and match, or sometimes I notice there's a glaring problem with one recipe. It's the same thing with science. You know, you want to review multiple protocols.
Yeah. So how do you make sure that you can mitigate, you know, as much as possible? Are there some simple things that scientists in Discovery Lab can do to mitigate it, or is it sort of not possible?
You know, one is more detailed protocols. I think that's important. Reagents are important. I realize that academia don't always have all the funding and try to create their own reagents. But when you buy reagents off the shelf from vendors, they're fully validated. If something goes wrong with any of those reagents, the vendors will hear about it right away. You know, so trying to use validated reagents is another area I think that could be helpful. And then perhaps more mentoring, reaching out to the experts. You know, there's a lot of expertise around, and seeking out those people, or more mentoring in the lab. But I think training needs to be improved with a more disciplined way of planning and executing your experiments, and powering your statistics is another area. You know, statistical analysis and the design of experiments is another area that needs to be improved.
Yes, it needs to be improved. Switching gears a little bit. So you said, I mean, you had the good fortune of actually working on a drug that ultimately your father took. Mm-hmm. Tell us a little bit about that, and how does that feel?
Oh, it's a privilege. It really is a privilege, and to me, I was very fortunate that this happened early in my career. Some people go through their whole career in drug discovery and not have that privilege, so it is a privilege. But because it happened so early in my career, it just really reinforces what you do and how you go about your work. And being rigorous and disciplined is critical in what we do.
Yes, you know, because it's a rough thing to come in every single day and go through this iterative process, and you could go on and on and on without, like, you know, having a single target that you've identified that goes on into clinical, leave alone into, you know, in a patient and in the market. So you have to have that commitment to, you know, to come in every day and go through this iterative process again and again and again.
There's always that, tomorrow we'll find that.
Tomorrow we will find that. Yes, we will find that. Well, this has just been a pleasure. I want to open it up to the audience because I know a lot of people have questions. So let's see. Do we have questions? Yes.
Hi.
Would you introduce yourself? Yes.
Hi, everyone. I'm Colin. I'm a PhD student in biomedical AI at Mount Sinai. I also run a biotech accelerator here in New York called Nucleates. My question is kind of around this idea in bioinformatics. We have what's called containerization. And so we put all of our scripts, all the data that we're using in a central thing so that it's easy and reproducible. My question is, how can we do the same for wet lab sciences, and perhaps maybe through avenues such as organ-on-a-chip devices where you can very precisely control which solutes you're using, or in something like Emerald Cloud Labs or Terra Biosciences where you now have sort of this digital control of the wet lab. Do you think that this can perhaps solve some of the issues that we're experiencing in the reproducibility problem?
That's great, great ideas. One thing that I'm noticing is just the notebook, writing down your experiments, and it's still surprisingly across many universities, it's still just paper. And when we work with academia, we try to instill in them using an electronic notebook. So I think this starts with some platform to be able to enter the information in, and a paper notebook where maybe you're not filling it out every day, it's not going to help you. So if you don't put the information in, I don't think there's any kind of AI or technology that would help, right? So I think the first thing is get the information in there, and maybe using more, maybe the academic institutions should put more effort in getting into electronic notebooks, because then you can leverage all this information. Yeah.
Yes, another question over there, and then behind over here.
Hi, my name is Paula Venines. I used to be a grad student and a postdoc. I since long passed it. I now work in marketing on the agency side. But I have a question about this notebook thing, and especially since you keep mentioning academia, there is, in my experience, what I've observed, and I can't imagine it has changed, an inverse incentive in academia. Because there's not enough funding. People, their mortgages, their livelihoods depend on publication, and it's not in their best interest to be transparent. Do you think that's going to be a barrier that we can overcome?
I think it needs to be somehow. I think it needs to be addressed. I mean, I can't speak for what academia should do, but this pressure of publication needs to be addressed somehow. And maybe a reward system should be different than just publishing. Maybe the reward system is somebody else replicating your data or bringing your ideas forward. I mean, somehow there needs to be a different way of rewarding PhD students, postdocs, and junior scientists trying to get onto a tenure track.
You know, that just sparked a question for me, right? I mean, gosh, how many peer-reviewed journals are there? Like, you know, it just feels like more and more are coming every day. So, I mean, what's the threshold? You know, I know that peer-reviewed journals, peers review the journal article and your results, but then, you know, what you're saying is that some of the data there is, you're not sure that it's reproducible.
So that's a really good point.
Yeah.
The peer review, so the journals are trying, you know, the Cell, Nature's, they're trying. I think you've seen the star, at the end of the paper, you have the list of reagents, even the lot numbers where, I mean, that's great. The star method is a great step forward, and the journals are trying something. They're trying to improve it. But I think the peer review is still weak. You know, some journals, you know, the peer reviewers are other academics who only, you know, who may know their area very well, but may not know other areas that is required to really address the reproducibility and go deep. So I think that's another problem as well, is just the rigor of the review as well.
Yeah. I think we had a question over here. Yes.
Hi, I'm Ajay. I used to be a bench scientist. In fact, the gentleman next to me was my colleague for several years. But then after that, I left the bench and I went to the computer. I happened to be a solutions architect for a company called TetraScience by day. And our sole mission is to do data provenance. The top 10 customers I work with are the top 10 pharma companies here today. But one thing is still true. Data provenance is an issue, not just in academia, but also even more so in industry. I have some answers as to probably why, but I'd like to actually hear your opinion. Why is data provenance an issue in industry?
Absolutely.
As much as it is in academia?
Absolutely. So in industry, it is also an issue. I've seen it myself. Just putting data in a notebook and being detailed enough. I remember when I was at Merck, I would search for data on a particular compound. We would have these big databases, and we could search for a compound, and we could see which assays it had been tested against, so which was powerful, right? So it's like, oh, it's been tested against all these 10 different assays, and the assays were given acronyms, like pizza. What is pizza? I could not figure out what that acronym was. Finally got to the protocol that was uploaded, and all it gave me was volumes of enzyme, mix with substrate, incubate half an hour, and read on a reader. I mean, that was as basic as that. I had no idea of concentrations, didn't even know what the enzyme was, didn't know what the protein type was. Very little information. And so so we tried to address that as well in industry, is improving the data quality that we capture. But it's not perfect. And again, it's this discipline of sitting down and actually writing it properly that someone else can actually understand. Because that's another problem, is that people don't realize that you eventually may not be there anymore, and someone else is going to have to read what you've done. And that's still an issue. And I think it's pervasive everywhere. I think it's just human nature that you get comfortable and you're not perhaps as disciplined in how you take your notes.
You could always tell who was a scientist and who was not by the notebook they carried with them. This lab notebook would come along, even if it wasn't an experiment, the notes even during meetings that they would take. But obviously, we all now have this and other ways to kind of just record, right?
Absolutely.
That should make it easier.
Yes.
Right?
Yes, absolutely.
Yeah.
Bringing your laptop or your phone
Yeah.
into the lab and taking live notes
Yes.
should be easy.
And hopefully that's becoming more of a practice for the next generation.
Hopefully.
Because I don't know that the next at least my son doesn't, barely can write properly with his hands, but he can type fast. Or can record on the phone.
Yes,
Yeah.
absolutely. That would be, if we can do more of that, that would be
More
great.
of that.
Again, the more data that you can enter in digitally, I think the more powerful it is at the end of the
At
day.
the end of the day.
There's a
Other question, yes.
from Tony.
Hi, everyone. I'm Tony with Deerfield Discovery and Development, and I have a question for Christine. And the question is, how do you think about the issue of reproducibility versus generalizability, and thinking about, how to deploy orthogonal assay formats as well as counter screening and confirmatory assays?
Yes. So one area that is challenging for everyone is when you're doing small molecule drug discovery and you're trying to find, you're doing screening and you want to find a molecule that binds to your target and has a certain activity on your target. It's really important to understand that small molecules can lead you down the wrong path. Like the Sertris example is a good one. The resveratrol, they thought bound to SIR2 and in fact it didn't. It actually bound to the substrate. So it's really important to not only make sure that that one assay is reproducible, but design other assays that can interrogate the mechanism in a different way to further get confidence that if your molecule's binding to your target, it should work in multiple different assay formats. And that's also the generalizability of your design is important.
Let's see if there are other questions. And I know, guys on the webinar, thank you so much for joining us. I know that's about 40 people are actually joining us virtually. And I don't think we have a way for you guys to send us questions. But if you do have follow-up questions, please reach out to us, send us an email, and we'll make sure to answer that. Anybody else in the room? Yes.
Eva Lawrence, Head of Biologics at Deerfield Discovery and Development. Christine, to what extent do you think confirmation bias may have an influence on reproducibility? Because, somebody may have a certain biological or therapeutic hypothesis, and then you interpret your results in a way that confirm that hypothesis and validate it. So how does that affect reproducibility?
Unfortunately, it does. And that's where you have to be rigorous in how you analyze your data and how you view your data. it's one thing to shine the light here, and there might be something there that's just important, and you need to take that into consideration. But that is, confirmation bias is an inherent problem in what we do, and it's understandable. Again, it's human nature. It's your idea, your project, your baby. You want this to be successful, but you also have to be aware of potential downsides or off-target activities. So yes, it is something that everyone should be aware of. And always, I mean, one thing that we're all very good at in science is just being very critical of other, when you read a paper, you're very critical. You should do the same for yourself. Be critical about your own work. Always question your own work. In drug discovery, the first thing I learned as a biologist is as soon as you start working with a chemist, they start criticizing your data right away. It's like, I thought my compound was going to be fantastic. How come it's not, super potent? And you're constantly having to explain, well, this is the data. So being critical about your own work is very important.
Good point. Other questions?
So the FDA recently passed FDA 2.0. That's part of the, the budget process that the United States Congress goes through. And in that, they enabled alternatives to animal testing. So, things like digital twins, things like organoid lab on a chip. So what are your thoughts on the reproducibility of that?
It's like what you said, right? Yeah. You were involved in the genome sequencing
Yes.
early on, and we thought you sequence the genome.
Yep.
That's it.
That's
Everything's
it.
done.
Everything's done. Check.
High throughput screening was the same thing. I think this is the issue with every new technology, and it just takes there's a big buzz at the beginning. I think there's a big hope, and then the reality sets in. I think there still needs to be a lot more validation. I don't think many of us are going to completely not do experiments in animals right now and just hope that this organoid on a chip will give us the answer. I think that it's great that we're going in that direction, and we need it because I think there is potential in that technology. But I think it's going to have its own sets of realistic issues that it's going to have to work out.
Yeah, I mean, whether you're, searching the computer for like a gene or a, SNP or something like that, or you're looking at the lab on a chip or whatever it is, at the end of the day, or putting notes in a notebook or dictating something, at the end of the day, I think what I'm hearing you say is that, science requires discipline and it requires rigor, and it doesn't matter whether you're doing it in a new system or an old system, that's what it takes to ensure that you can reproduce the results.
Well, but it's also more training, understanding the intricacies, the technical challenges, so that you could write all the notes and everything you need, but if you don't understand fundamentally every aspect of what you're doing, it could also lead you in the wrong path. I mean, a lot of these things is just inadvertent or perhaps even ignorance. You really believe in your data and you've done everything proper, but if you missed a really significant step or you don't understand a basic function, it's just going to carry through until someone else who's an expert will see that and figure that out.
In the old days, students worked very closely with their mentor, and there was a sort of like an apprenticeship method to teaching, students, and that's gone. So like learning how to do an experiment on YouTube doesn't work, right?
Well, you can peruse YouTube and get some ideas, but no, I think that's another aspect of it is having a mentor in the lab, your supervisor, help you out, or at least guide you where you can get the information. I think that's very important. Sometimes with big labs, everybody's busy. It depends also on the leadership in the lab as well. Sometimes you may have a big lab, but the students help each other out. Everybody learns from each other. And I think science is a collaborative effort.
Yes.
And the more you reach out, the more you learn from your peers. I think the more success in the end of the day.
Yes.
Yeah.
That's great. Okay, we have time for maybe one more question, I think. Jamila, keep me honest. We have time for one more question, one last question.
Nope.
Yes, there you go.
Hi, yeah, thanks for a really interesting discussion. Sorry, yeah, my name is Jonah. I'm a postdoc at the New York Genome Center. So yeah, I guess like coming from an academic perspective, I'm just sort of curious what you think would be a sort of ideal symbiotic relationship between industry and academia. Maybe I'm pushing back a little bit because I feel like this talk is very critical of academia, which I totally follow and understand and sort of get where you're coming from on it in a lot of ways. Yeah, but I'm just sort of curious, like you said in the early 90s, a lot of the times the industry was actually doing the bulk of the research and academia was a little bit behind. But now that you have maybe competing objectives and incentives on both sides, like how would research from academia actually positively impact industry with this reproducibility issue that comes up so much?
Yeah, I would encourage more partnerships. We work on programs where we have an academic lab that we're collaborating with. We have postdocs working in the lab, and we really enjoy that collaboration, but also making sure each side understands what they're doing. It's a good way of learning from each other. And I think if we could have more of that, because then on one hand we get to see what your challenges are, but also you have ideas and thoughts that are perhaps outside of our wheelhouse or perhaps outside of our reach, and we welcome that. And then on the other hand, the academic side learns why we have to do certain things in a certain way and why we have to be repeating the experiment 10 times before we're ready to say that we have an assay ready to go. So it's this learning from each other, and it's not easy. It's really, sometimes there's a wall dividing the two enterprises, and you know, the more we can break down those barriers and learn from each other, ideally, if there was a way of getting internships in, you know, I think in pharma we do have the internships within the industry. That's a great way of learning. But I agree, and again, you know, we've been talking about academia, but because the literature, you know, pharma do publish, but it's not our bread and butter to publish. But it is a problem even within industry. We have similar issues, and perhaps a bit different, but this problem is pervasive everywhere.
So we want to end the evening on a positive note, right? So the problem is pervasive, but there's a lot of people working on solving it, right? Especially led by the NIH.
Yes.
So closing thoughts on that?
Well, again, it's awareness, being open to the issue, and the NIH is very much trying to improve that, and just understand that we're not being hypercritical, we're just trying to improve the science and having access to the NIH training. You can go online, there's some trainings that you can follow. But you know, getting more mentorship, more understanding of statistics to power your studies. And I think everything's out there. It's just a matter of actually following through.
Yes. Following through and, you know, taking the time and the discipline and the rigor. that's involved in, making sure that, you're on the right track.
Correct.
Right? Yes. Well, thank you, Christine. This was really, really enlightening. Really appreciate it. And we still have some popcorn and chips and wine. So if you have more questions for Christine, you're around for a few minutes more. Yes.
Absolutely.
Thank you, everybody. Thanks.